Hello, welcome, and it's great to see so many of you. And it's really good to be back and joining you for another Reach Out Rachel webinar. And boy, is it going to be a webinar. We've got a really interesting session lined up for you today. So we are delighted to be joined by Dr. Anna Sadnicka and Dr. Hortensia Gimeno, who have kindly agreed to share their insights from their work with you today. We're so excited to have you both with us. So thank you very, very much for joining us. So tonight, Anna, if it's all right with you, we will be kicking off with you. Dr. Anna Sadnicka is a consultant neurologist at the National Hospital for Neurology and Neurosurgery and a Wellcome Trust early career researcher at Gatsby Computational Neuroscience Unit. And her award-winning research combines behavioral experiments, electrophysiology, and computational models to shed light on dystonia mechanisms. Anna, did I get that right?
Perfect.
Lovely. In which case, I'm handing over to you, ladies and gentlemen. Anna.
So it's my great pleasure to try and give an update on lots of current research that's going on the role of the cerebellum in dystonia. So the first thing I wanted to do is just to introduce the cerebellum a little bit. The word itself means little brain, and it's this kind of nugget at the back of the skull, shown here in brown. And it's a pretty remarkable structure. So it has more than half of the neurons, the cells that do all of the work in the human brain. And it consumes a fifth of the total brain energy that the brain requires. This is another animation that just shows you some of its beautiful complexity. It's a highly folded structure, and this kind of shows you how, if you expand it out and down again, you have this huge surface area over the cerebellum. Here, the idea is that you have a very large surface area, 80% of that of the cortex of the brain. We believe it's doing many things. In humans, in recent evolution, this is the part of the brain that has really expanded compared to other animals in the animal kingdom. So we think it has very important roles in human function. So what does it do? We've got beautiful neuroscientific methods now that start to unpick this a little bit better. So now we've got a different view of the cerebellum. This is a little bit like a flat pack furniture. So we've just folded it all out, squashed it onto the page into 2D. And I think everyone will be aware that the cerebellum has roles in movement, but it's also important for a whole host of other things. So you can see some of them listed here. Eye movements, attention, mathematics, language, memory, emotional processing. There's a lot of things that the cerebellum is doing that we're becoming a lot more aware of. So why the cerebellum and dystonia? Now, like many very promising areas in research, this kind of whole region of research has spawned from observations in patients. So if you have, for example, a lesion in the back of the brain, the posterior fossa, such as a tumor, this can present with a dystonia. Equally, if you successfully remove that tumor, that can lead to an improvement in dystonia. And so here you have this nice kind of cause and effect relationship between something happening in the cerebellum and the dystonia as a symptom. And we also have patients that have other disorders of the cerebellum. They present with symptoms that are suggestive of the cerebellum, but they also can present with dystonia. So again, these are just more clues from the clinical room that there's something important about the cerebellum. So researchers have tried to take this forward and use lots of different types of experiments to do this. This is now example from animal experiments. This is a mouse. And interestingly, if you inject a chemical that excites the cerebellum, this one's called cyanic acid, just by exciting the cerebellum alone, that's enough to generate dystonia. Alternatively, if you have bred animal models of mice that have episodes of dystonia, if you remove the cerebellum, this completely abolishes the dystonia attacks. So again, we're seeing evidence of this cause and effect between the cerebellum and dystonia. Why could this be important? Well, we need to develop new ideas and understanding the mechanisms of dystonia, but also new treatments for dystonia. And we've known for quite a while that the cerebellum and trying to modulate its behavior may be very important in humans. So this is a article from the 60s by a very early neurosurgeon, Cooper. And this is a patient with a neck dystonia that's twisting around to a particular side. And I don't know if you can see these muscle traces here. They're very, very busy with a lot of muscle activity. So this is an active dystonia. Look at this post-operatively now. Can you see how flat these lines are? This is after a lesion to the thalamus. And the thalamus is another brain region that sits slightly further away from the cerebellum, but it's an important outflow for all of the cerebellar information. So you're essentially blocking the output of the cerebellum. And by doing this with a lesion in the thalamus, you can improve dystonia. But there was a pretty major missing part of this puzzle, is that we still don't have very good understanding in how the cerebellum is interacting to cause dystonia, what the mechanism is. And so what I wanted to do was to slowly go through one experiment that we've done to try and address this, and then to discuss what sorts of ideas this gives us about how the cerebellum might be involved. So this is quite a famous neuroscience experiment, and some of you may have heard of Pavlovian conditioning. It's most famous in the context of dogs, so I'll just take you through this. So like many of us now, we might be a bit hungry. If we see food in the horizon, we maybe start salivating. So this is a stimulus food. It's unconditioned because we just naturally, unconditionally start salivating in response to the food. Before the experiment, if you hear a bell ringing or the dog hears a bell ringing, this is a neutral stimulus. There would be no salivation in response to this. But if the bell and the food are repetitively paired, the dog learns that association. And you can start to get, after repeated pairings, salivation in response to the bell alone. So this is called Pavlovian conditioning. This is a fascinating experiment because you can test it across species. So this is in a dog. What does it look like in a human? So here is the setup in a human that you can do in an electrophysiology lab, which was one of those long words at the beginning. So you can put an electrical stimulator on a nerve, the supraorbital nerve, and that just makes the eye blink. And that's unconditioned. You're stimulating the nerve, it makes the eye blink. At the same time, before that eye blink, you can put a sound into headphones. This is now our conditioning stimulus. And we're going to keep pairing these until the human learns that sound is anticipating the stimulus which causes the blink. What does that look like when you're trying to examine whether or not this is occurring? Again, we're looking at muscle activity. This time we're looking around the eyes. So here, this is before a response has happened. Here's the auditory tone in red, nerve stimulation, and then we see this muscle activity. After lots of pairings, can you see here that we're starting to see the muscle contraction occurring before that nerve stimulation? That's all an unconditioned responses. And so you do repeated pairings and you can count the number of conditioned responses that you start to see. We don't need to look at the detail of this, but I just wanted to flag that because we can examine it across species, it is remarkable that different elements of this response we can map to individual cell types in the cerebellum. So you're doing something in a human in a lab that's non-invasive, and yet you're beginning to get some insight into the actual responses of the neurons within the cerebellar circuitry with all the different types of cells within the cerebellum. So it's quite a powerful tool. So this is eye blink conditioning results in humans, and there's been quite a number of studies published, but the difficulty was that we had quite low numbers, and the results were, yes, it is affected, maybe it's affected, my study, not really. And so this was quite a confusing kind of horizon of results because it was difficult to know what exactly this meant. And I think this was because we had very small numbers and we needed to pull this data together. And so that's what I want to show you the results of. I want to show you the results of taking all of the data from these original studies, putting them together. So we've got 50 patients with dystonia and 50 controls. Now, I'll just take you through this plot. This is all of the controls, okay? So on the bottom, we've got block number 1, 2, 3, 4, 5, 6. Here, we're just counting the number of conditioned responses we have, and we're saying a percentage of conditioned responses in each block. And then along here, we've got the number of different individuals. So each of these lines is a different individual. I've ordered them according to whether they were poor conditioners or good conditioners. Can you see down here at the bottom, we've got lots of individuals that are just hugging the 0%, people that aren't conditioning at all, whereas others are very good conditioners. And even from the very first block, they've got 75% conditioning. They go on to have near to 100% conditioning responses. So that's the range that you see in the healthy population. And if you look at individuals with dystonia, you see a very similar range. This is quite significant variability. We know lots of things influence the rates of conditioning. Look at this, you can split them by age and you can see them kind of fanning out here. You can also split people by sex and you can see that females are slightly better conditioners than males on average. But interestingly, once you've collapsed all of these results together, dystonia and their age-related, age-matched control group are very, very, very similar. Now, is this, you may ask, because we've collapsed all of the different subtypes of dystonia? Well, actually, even if you do a more subtle statistical analysis where you're subdividing the subtypes of dystonia and kind of accounting for the numbers of dystonia you've got, you can plot the controls in this with this grey line here. And for any of the groups of the dystonias where we've got reasonable numbers, there's very, very similar rates of conditioning. So this was quite an interesting result. And I think it really does reflect the technique that we're using. It was very, very variable. And so you could use this to say, right, well, if we wanted to detect a 5% difference in our patient group, how many subjects would we need in an experiment in the future? Look at this, it's 700. Could you see that there's original studies had very small numbers? And so, you know, this is revealing quite a lot about the way we're doing these experiments and what they can tell us. So if we just think of a summary of this data, overall, eye blink conditioning as a proxy for cerebellar function, as a proxy for what all these different cerebellar cells are doing, looks pretty intact in these isolated dystonias. We can't really see a clear abnormality. Some of the data that said this is firm evidence that the cerebellum's involved, it's not there, that data. And so, you know, this is seemingly at odds with this expanding interest that everyone has with the cerebellum in dystonia. And what I think this data is revealing to us is that the cerebellar role is likely to be more complicated than something we can pick up on with this sort of paradigm. So this is kind of the part where we say, well, what could the role of the cerebellum be in dystonia? So this could be a cerebellar disease. Have we got it all wrong? Were the other structures we were thinking about nothing to do with dystonia? In the most simple model, this would be an on-off switch. If the cerebellum's not working, you get dystonia. Well, I think just through the discussion that we've had already, you can see that this isn't a very good model. We know that there's big differences between cerebellar disease and dystonia. It's clearly not as simple as that. So if it is a cerebellar disease, we clearly need other kind of parameters, other things that are measuring how the cerebellum is going wrong. Is it a loss of function or a gain of function? Is it a small part of the cerebellum or all of the cerebellum? These are the sorts of things that people are debating within the literature. Alternatively, is it more of a discussion between two nodes within the brain, the cerebellum and the basal ganglia? So the basal ganglia traditionally has had a huge role in dystonia. And is it actually that the cerebellum and the basal ganglia are talking to each other? If that is the case, we do need to still have more information about what is going on and how it is that these two dysfunction in these two regions are causing dystonia. But then many of you may have heard about the network hypothesis of dystonia. And here it gets even more confusing because we're getting lots of other different regions, the cerebral cortex, the thalamus, the basal ganglia, the cerebellum, the spinal cord. How are all of these different regions talking together to contribute towards dystonia? But that's not all, because actually we know that we have kind of genetic influences for dystonia, but we also know that many subtypes, particularly the focal ones, have influences in the environment: the dry eyes before blepharospasm, the overpractice in musicians' dystonia. There's many influences in the environment, and we need to understand better how these integrate with the dystonic network to cause dystonia. And then the thing that Hortensia is the expert on: how do these changes within the brain develop over time? What does a brain look like when it's vulnerable to dystonia? What does it look like when dystonia starts? And what does it look like after dystonia has been present for a number of years, after the brain has been trying to offset the dystonia? So these are lots of questions that we are kind of all working on in the background, using animal models, human methods to try and better establish. But there are some pretty amazing early clinical results even without this knowledge. So here's just 3 studies that I've just cited. And what are these doing? These are already in humans trying to stimulate the cerebellum, or the nuclei, the centers within the cerebellum, to see whether modulating its activity helps dystonia. The first thing is obviously to establish that it's safe, the methods are safe, that we have familiarity with the methods, but it's also then to look at what it's doing to the dystonia, which subtypes of dystonia it's helpful in, and how it might be working. So there are early clinical results that kind of back up this literature that I've been discussing, and some of the questions that we need to answer in terms of where this could be going in the future. So in conclusion, I've hopefully kind of introduced you to the cerebellum. It's quite a hot topic in the field of dystonia research at the moment. It's a very complex, beautiful structure. It has many roles in humans, and it may well have a very important role in dystonia. We've got methods now in neuroscience that are really helpful, that can actually help allow us to look at the cerebellum in new ways. And we need to get more of these methods across into dystonia research so that we can compare healthy controls to the dystonia population. I think we've come a huge way, particularly with this research, not understanding that well exactly what the mechanisms are, to be starting to develop treatments already that may be effective. But where I hope the real power is if our research actually starts to tap into these mechanisms and we understand it more. This will be the real key to therapeutic innovation and breakthroughs. And I am incredibly hopeful that in the future, many subtypes of dystonia can be more effectively treated. And I think this is particularly with methods such as neuromodulation and neurorehabilitation, because we have this beautifully dynamic disorder. Often there are no lesions within the brain that we need to offset. And so if we can really understand the mechanisms better, I do think we're going to get much more effective at being able to offer treatments to people with dystonia. So I was trying to bridge a little bit there onto Hortensia's talk, because I'm like a huge follower of her work and the power of the neurorehabilitation in dystonia. But that's the end of my slides.
Thank you, Anna. That was really interesting and put beautifully. So thank you very much. We will have time, ladies and gentlemen, to put some questions over to Anna in a bit. But first, we're going to hand over to Hortensia. So Dr. Hortensia Gimeno is a clinical academic occupational therapist and works as an Associate Director of Therapies Research at Barts Health in East London. She is an honorary senior lecturer at Queen Mary University, London, with a strong ambition to improve the care of children and young people with movement disorders. Hortensia, over to you.
Thank you so much. Thank you so much for the invitation, and thank you, Anna, for that lovely plug on how to follow with a very different talk. So I'm just going to share my screen, and as Anna mentioned, my work is on rehabilitation, And before I start talking about dystonia and the cerebellum, I've got a big disclaimer because I'm not an expert on the cerebellum, and obviously I know where it is in the brain, and it's actually the only structure that I dissected in a lab when I was a student many moons ago. And obviously, when I started working with childhood onset movement disorders, the talk was about the basal ganglia, and I know the cerebellum came a few years after. So that is one of the disclosures. And the other is that a lot of the work that I have done is in with children and young people with dystonia and other movement disorders. But I think some of the lessons, if you like, of what we've done in rehabilitation apply to anyone with a movement disorder. So that's a little bit of a context. And in childhood disorders, we talk about hyperkinetic movement disorders because sometimes there's a few other things going on apart from dystonia. And we do have a lot of genetic and rare conditions as well. And but the largest group that we have in children and young people is dystonia in cerebral palsy. So that will be our largest group. And I'm sure you know about these. And one of the things that is really important for me, I'm just going to mute those. One of the really important things when I was working clinically at the Evelina with, and you might know Jean-Pierre Lean, I was working in the deep brain stimulation service there, is I was extremely frustrated that I didn't know how to advise people on rehabilitation after deep brain stimulations because there was no evidence, there was no studies. And even if you look at the definition of dystonia, and I've highlighted there in blue, so I'm an occupational therapist by background. My job is to help people do the things that they want to be able to do, right? Whether it's a child that wants to pull their knickers up, play with Lego, handwriting, whatever it is. And often, and the problem is that the more someone with dystonia tries to do those things, sometimes the worse the dystonia becomes, which is a problem for rehabilitation. And that is one of the issues why there's not a lot of studies out there, and how many children with dystonia and cerebral palsy even have been excluded from research studies. So the treatments are varied. So we've got obviously the pharmacological interventions, and we've got deep brain stimulation as treatments. Even those are low evidence. We don't have a lot of studies. And I think some of the problems that Anna was talking about, the smaller studies, we can't really make big conclusions. And obviously, this is not what we do in rehab, but it's one picture to exemplify other approaches that are not pharmacological or surgical. And really, we don't have a lot of evidence. My studies probably were amongst the first studies, and they were in like 2017. But I wanted to show you some of the clinical work data that I collected, and this was thanks to some of this data I've put together thanks to a small grant that we got from Dystonia UK. So I thought it was only relevant to show you how we use some of that money. And I'm hoping that we can finally put this into paper format. And what I did was this assessment that looks at, so children, young people, I bring them into the kitchen, or and then we do two activities that are part of an assessment that is activities that have gone through a lot of validation. And then you can see three groups there. So the acquired dystonias will be largely kids with cerebral palsy. You know, we've got quite a few genetic disorders, DYT1, DYT11. We probably don't talk about DYTs anymore, but they don't roll out of my tongue, like the new terminology. And then we've got quite a lot of young people that actually we don't have a diagnosis for them, and they might have genetic disorders, but we don't have. So that's the idiopathic group. And this test looks at your motor skills and your process skills. So your motor skills will be around trying to reach for something, trying to use two hands together, whether your hands are really shaky or not. And your processor skills look at your ability to problem solve when you're doing something and to organize and plan. So for example, just to give you an example, if somebody's frying an egg and then your dystonic arm is pulling you away and then you've got the toast on as well, can you actually figure out if the egg drops? Can you figure out what you problem solve at that time? And obviously it is quite difficult when you've got dystonia pulling you away from the task. So, and then what we did is we looked at, and this assessment takes into account where you should be for your age. And this is the red line. Below the red line means that you do have motor difficulties to do those activities. So you can see everyone is below the motor or the process skills. And the really interesting thing is when we did DBS, so you can see that everyone has improved, but the children, even with the genetic conditions that do really, really well with deep brain stimulation, they're still below where they should be. And these are the kind of young people that were coming to me and saying, H, my dystonia is great, but I still can't catch a ball on the playground, so nobody will play with me, or I still can't carry a bowl of cereal. And this is what kind of got me. I never wanted to be a researcher. Anna probably wanted to be a researcher from day zero. I was just like, I'm a clinician, but I was so frustrated that I didn't know how to actually help children after deep brain stimulation, that's what got me thinking about my PhD. And then for the process skills, you can see the same. You can see that actually here we've got a little bit more. This is a 2 years, so it's a baseline, 1 year post deep brain stimulation and 2 years post deep brain stimulation. So you can see people improved after deep brain stimulation, but there's still some kids that do need a little bit of help. And I think this also speaks to the need for us to do assessments that are meaningful to patients as well. And just to show you, to illustrate this point, so this is the Berg-Van Marsden dystonia rating scale, which you probably are familiar with. And it looks at more of like the impairment and how much dystonia it is. And what happens in children and young people, because obviously it was developed for adults with dystonia, is that when we applied that assessment to children, even though they improved, these are the same children that I've shown you the improvement on the other assessment, we're actually not able to capture that change before and after DBS. We can on the genetic group, but we can't so much on our largest group, which is the cerebral palsy group. So with that, I decided that I wanted to try out this approach called the cognitive orientation to daily occupational performance. And this was developed for children with a different type of condition that, and when I was seeing my kids with genetic disorders, and I'm not saying they have the other condition, but they looked a little bit like the kids with developmental coordination disorder. So there I went to get some funding to be able to do this. And we did co-op after children have had DBS. So I wasn't, I didn't decide to be a scientist from from the day I qualified. I want to be a clinician, but it was very much these patients talking to me about the need for something else that kind of drove me to to get going with fellowship applications. And we did this with a fellowship from the NIHR. And the co-op approach is based on a learning paradigm. So it's looking at thinking about the way we think about problems. And I think this is relevant to what Anna was saying in terms of, how do we learn? And it's going to be really good to talk to Anna about these mechanisms or what we think the underlying mechanisms are, because I can show you some of the videos before and after. And this intervention is not about hands-on. I don't do anything. I actually don't do a lot of the work. The kids or the patients do most of the work. I help with asking the right question at the right time. And somebody put it as is something to achieve whatever it is that people want to achieve, those goals. So we focus on whatever the person wants to try and work on, and we use something called cognitive strategies to reach these functional goals. So there can be very different types of goals. And the idea is that the person learns to problem solve so that they can apply these skills to other things that they don't work in therapy. So this learning paradigm is really important, and it's a paradigm shift. So a lot of rehab is being potentially based on other things where the therapist might be doing something to try and reduce dystonia. We don't focus on the impairment. We focus on the one thing that the person wants to learn. So this, you might have seen this video already. So this is a girl that wants to
and then you just
learn to ride a bike. A bit out. So she wants to learn to ride a bike. This is how much she can do before
even if it is with
the intervention. And she's been trying to ride a bike for 10 years. Just going to stop it there. And this is how much we were able to do with 10 sessions. We worked on 3 goals. And she wanted to be able to ride in a straight line.
Good.
And this is what she was able to do. But obviously, nobody rides a bike. Nobody rides a bike for 10 meters, right? So when I went back, and this is the really important thing, when I went back 3 months later, and we hadn't done any more treatment, this is what she could actually do. And I think this is the real key. Without any more intervention, she was able to ride the bike for much longer, but also she was able to be part of the group of kids that, family members. She was able to, she was much stronger on her core because she was doing something that she was really motivated. So now she could ride the bike. Her involuntary movements probably scored the same on the involuntary movement scale, but she was able to do much more, and she was able to think through any of the problems that she had with other activities. So I'll just show you, I show you a couple of the quotes that she gave, and she presented at a conference with me and with another young person. And for her, it was about how much confidence this had given her as well, and how she often describes it as other people didn't think she was going to be able to do any of it, and she actually showed everybody wrong. But these kind of stayed with me. And Gareth, who's the other person, I'll show you later on how he explained the intervention. But I just wanted to ask you, and this is pertinent, in terms of how we got her to ride this bike. Obviously, dystonia was one of the things that she had a problem with, but I don't know if you know who this guy is and if you know anything about him. I don't know if you want to put in the chat. I can see some people smiling. So it's Michael Phelps, and then he's a very good swimmer. And I don't know if you know anything about him, but he's got a lot of medals, and he's got very long limbs, and he's won a lot of medals. And then sometimes in some Olympics, he won even... he beat his own record, and a lot of people did these. And I just wanted to show you these to try and illustrate the importance of different things in rehab, or different things that we think about. We call it system models. So for example, dystonia will be one of the things that Grace on this video is part of who she is, but so is the environment. So she had a very supportive environment. Her brother went out with her to do bike riding all the time. She was extremely motivated, so that motivation to prove everybody wrong and to do bike riding. She was extremely bright, so she could problem solve what she was doing, and that is all part of the puzzle of how we learn new things, or how we relearn things. For people with a stroke, for people with dystonia, or for people with other conditions for whom we've used this intervention. So this thing of practice makes perfect. I tend to challenge that idea, because if what you're practicing is the wrong thing, if what you're practicing is not being able to do something because your dystonic arm is pulling you away from frying that egg, that's what you become good at. So instead, what we do in this intervention is we do something called goal, plan, do, check, which is a metacognitive a strategy to think through things on what is it that we're trying to do, and then we develop plans, and those plans or those strategies is what people develop. And it's not about normalizing movement or doing it in the normal way, whatever that means. It's more about finding ways for that person to do things. And this is how Gareth talked about it. And for him, it was really important to be independent. He learned how to cook pasta, and he explained it to the audience that he was able to then use those strategies to dampen down some of the involuntary movements for when he was driving a car. And that is what we call transfer of skills. And he talked to the audience about how understanding where things were breaking down helped him to then figure out what he needed to do about it. And in turn, what this gave him was confidence. And by gaining confidence, his movements were better. So we didn't work on the movements, but the movements got better by him developing more kind of self-efficacy, knowing that he can solve his own problems. And this is a story that we've heard from quite a lot of the people that I've worked with. So really the idea, and I had a bit of a poll, but for interest of time, we're not going to do this, but I'll share my email address at the end. But the idea is, as Anna said, we did this with a few numbers, but really we need to be able to do this on a randomized controlled trial. And I'll show you some of the issues that we have in running a randomized controlled trial, and in children and young people, and I don't know, in other conditions, is that they tell us, well, sometimes I can do something really well, but then I can't do it again. And we've got this unpredictable and this variability of movements and ability to do things. So if I show you this video, what we do in clinical services and what we do in a lot of research is we assess people at one point, we give the intervention, and then we assess again. And if you've got that level of variation and fluctuation, is really difficult. So this is somebody trying to do the eyeliner. And this young woman is a really bright young woman that wants to go to university. And you can see that she's trying to do the eyeliner. And this is before we gave the co-op intervention, and this is after, and I'm not going to play the videos because they're a bit long. She discovered her own strategies, and this is what you can see before and after. So this is with dystonia myoclonus, and this is how she was able to do it. If we only do those 2 data points, we will have showed that our intervention failed. Right? But you see the videos, you look at the videos, and she is much better, right? And the problem is this variability that you see here. So this is all before the intervention. So sometimes she's better than other times. Sometimes her movements are better than other times. And you can see here, post-intervention, there's still variability, but it's much less, and she's much better at it. So really, we need to think about how we capture these on any clinical trial, because obviously one data point might not be what we need to do for people with dystonia. And this is just, you can see the change in the slope. So these are some of the strategies that people use. And I'm not saying go ahead and use these strategies because this is what we worked. What we did is categorize these with the students that looked at thousands of hours of videos. And things like distraction helped, where the focus was in terms of attention, relaxation, imagery. And these are things that are starting to make us think about, how do we do these? And for some people, there were some strategies, and for other people were different strategies. So we probably do need to think about how we test these in a bigger trial. And you might yourself have some of these strategies that help with dystonia, but these were in children and young people. So we don't know exactly when to, for example, introduce distraction as a strategy, and how to guide for that. So really, I guess what I learned from these studies and from working in this way with children, young people, is that probably a lot of what we do in terms of motor performance, in the things that we do day to day, are mediated by cognition much more than we originally thought. And I guess what we've done is ask more questions than giving answers. But yeah, that's where we are. That's my email address if there's any questions. I'm sorry I've taken quite a bit of time, but I think we've got a bit of time for questions.
You have not taken quite a bit of time, Hortensia. Thank you so much. It was really, really interesting. We do have a couple of questions. Anna, I think probably I'm going to throw over to you for this first one. This question comes from someone who says that I have cranial dystonia or Mage syndrome, and their sister has just been diagnosed with cerebellum degeneration causing ataxia, and they ask if there could be a genetic link.
Yeah, I mean, as you were saying at the beginning, it's always tricky to comment on kind of specific cases when you've just got a very kind of small summary.
Yeah.
I think the answer is yes, there could be a link, because your sister has a genetic disorder, and so therefore there could be, but equally, you know, occasionally rare disorders do occur sporadically. They come out of nowhere, and so I think it's a very interesting discussion to have with your clinician, but it's not a kind of, you know, I was trying to just debunk in that talk some of this kind of certainty that people seem to give between the cerebellum and dystonia. There's so many questions still, and so you know, even if I think there could be a link, but there's certainly no kind of definite reason for the Meg syndrome to be caused by a genetic cerebellar disorder, and so have that discussion with the clinician. Yeah.
Brilliant, thank you. We've been asked if can any cerebellum involvement in dystonia be seen on a brain MRI or by any other tests at the moment? Either pick which one of you would like to answer that question.
Yeah, I mean, again, it's really going into the experimental methods currently, and so hopefully this is where the research would go in the future. Because my research has certainly drifted away from this sort of thing, because I am also quite pragmatic, and I want to be doing what Hortensia is doing, but I want to be capturing the before and after. I want to be capturing the complexity of some of the change that people are doing, and so we're going to need these more advanced methods so that we can look at activation patterns with something like a functional MRI, where you're looking at activations, you know, before and after, maybe even some of these mobile methods, so we can look at brain activity now where people are freely moving, where the sensors are wireless. And so I'm really excited about the fact that we can hopefully look at the brain in ways that's actually more clinically useful, and properly look at how these interventions that are very complex are having an effect, so that there's more of a dialogue between therapy and the science that needs to catch up and provide the evidence of what they're doing. Yeah.
No, absolutely, and brilliant answer. Thank you. I think it's quite exciting hearing about things that are potentially coming up and how that will affect the community. Our next question comes from someone who says they have stage 1 cerebral palsy and found out they have dystonia. They're an OT in their work, and their balance means they can't ride a bike, but most of dystonia affects their jaw and face, and have used problem solving skills throughout their life, and I'm very impressed with the information. So thank you both for that. Their question is there any research focused on growing older with dystonia?
In terms of the like longitudinal studies that
I think so. I think obviously the your presentation was largely because that's your area focused on children. Is there something similar happening but for for an older age group?
I mean, I'm in adult, so I guess I should try and answer that question. So I think, like, overall, although there is much variability, day to day, in terms of the overall pattern, people always say that dystonia is interesting because it comes on over a few months and then it has a relative plateau. Yes, there is some fluctuation over it, but there's a relative plateau. And so it's always fascinated me what that equilibrium stage is. What is it? It's something has happened within the brain that's caused the dystonia, but then it seems to stop spreading, or it seems to reach a pattern of stability where there's a relatively stable disability. And so yes, there's lots of research into why that might happen. Certainly there are changes as the body naturally ages, and so I think to kind of pad that answer out a little bit more, as you naturally age, you can see changes that happen with the dystonia because your nervous system is changing as you age, and there's new diseases, or there's other things that happen in your life. But yes, I think people are thinking about this, but again, there's lots we don't understand.
Brilliant, thank you. It's nice to know that that's going on. You spoke about clinically useful interventions, and obviously many of our audience today are living with dystonia. What kind of timeline should people expect to see advances in treatment? You said you were feeling really optimistic, but obviously optimistic from a researcher's point of view is slightly different to be able to go to the doctor today and get the treatment tomorrow. So realistic guidelines, I guess I'm asking for. What should people be looking at, or how long?
Well, we need to be able to clone Hortensia. That's the first thing, because it's
Hortensia needs to get funding.
Yeah, because one of my huge frustrations as an adult neurologist is that I have limited access to the sorts of therapy that I wish I could deliver, and a lot of that is because the argument is that we don't have some of the research to back that up. But it's also great problems with the infrastructure, and so I think that's something that both of us are huge advocates for, is just to really try and bring the services forward in that way. And so yes, I think these sorts of initiatives should be coming into patient care in the next few years. I mean, Hortensia is already doing this, but these sorts of methods have utility in adults as well. We need physiotherapists that are specialists just in dystonia. There's so many types of dystonia. So I think we have knowledge already that should be better implemented within the NHS, in my opinion. And then there's lots of new things that should be flooding on. But yeah. There's lots to do, and a lot of it is funding as well that needs to kind of get into these sorts of initiatives, I think.
Is there any opportunity, so for the people that are on here today, or Hortensia, you mentioned sort of wider audience getting involved in research, how do people want to do that? Sorry, not how do people want to do that, how can people do that?
Get involved in research. Yeah, I think so this is really interesting, because we actually I do think that funders, particularly the funders that I will apply to, which is very like clinical work, trying to reach patient benefit quite quickly, what they're interested is in the patient's voice, and interested in people with lived experience, and what the priorities are, and what should we be researching. And so I think there is an opportunity for Dystonia UK to lobby some of the funders for more kind of research that addresses the priorities of your membership. So that's one way. The other way is and this is a small plea, probably Anna also needs, like we run things like patient and public involvement with funding associated with it. So I certainly, if I get the funding that I want to get to ultimately run a trial, I will be putting together a panel or an advisory panel and people to work with people with lived experience, and that will be children and young people and families. I think we have learned from people. So to your question around, how long does it take for interventions to be adopted by the NHS? Or the gap between research and implementation in clinical services is 20 to 25 years. And I don't think it's ethical, and I don't think we've got that time. And there are different strategies and different things that both NIHR and NHS are trying to do to try and get implementation to narrow that gap, right? We can't have patients waiting for 20 years, but for anything to get picked up on the NICE guidelines, for anything to be picked up on clinical guidelines, we need the research behind. And for that, we need the big clinical trials, the big randomized controlled trials, which are problematic when you've got loads of rare disorders, when you've got variability. And this is why we need patients to actually design those trials with us to see what's feasible. So that's how people can get involved. That's some ideas. We can run a research workshop with you at the next conference, maybe, to get people involved.
Yep. Right, I think we've got time for a couple of very, very quick questions just left. This is one for you, Hortensia. Someone has asked, they were wondering if you modified the co-op approach at all for children with cerebral palsy in comparison to children with developmental coordination disorder, for example, particularly those with different levels of cognitive ability. Or does it look like it's most effective for children who have very good cognitive ability?
So I did 2 studies, and the approach is the same for that is being used before. I think the key is actually how we train therapists more than how we deliver it. So we did 2 studies. The first one, I was the OT for all of the kids. And obviously, I know the approach really well, and I know dystonia really well. So I think what happened is that I was much quicker at helping the kids identify the breakdowns because I knew dystonia really well. Then the second study, we had 7 therapists trained on the approach. And the fidelity, so how well they were delivering the approach, varied. So some people were amazing, even some newly qualified. So not very experienced, but they were amazing. Some people less amazing at keeping to the approach, because sometimes the more experience you get, the more ideas you have about what works and what don't work. So the work we need to do is around that fidelity and how we train people and the cognitive ability. We actually have individuals on those studies with some intellectual disabilities, so mild to moderate intellectual disabilities. And these are two of the questions that I want to do on my next bit of research if I get funding, is how do we include those children with intellectual disabilities that might be associated to kids with cerebral palsy. Can the approach be done with them? Can it work? And then basically a series of feasibility questions so that we are set up to run a trial in a few years.
Brilliant, thank you. And I think just in the objects of time, because I think I could talk to you guys all day. I think myself and the audience find all of this fascinating. But an interesting question for you both to end on. What's the most interesting thing that you've learned about dystonia in your time studying the condition and working with patients? Anna, do you want to take the first hit at that?
It's a slightly frustrating answer because I just feel it's endlessly fascinating in the sense that we've just not fully got a grip on what is going on within the brain. So I, as my job as someone who wants to try and understand something better, dystonia has got me in every way, because I just think there's so much more that we need to understand, and also so much more that we can do. So it's a little bit of an unsatisfying answer, but it's the honest one, in that it's, I think there's a lot of work to do, and hopefully we can help contribute to that story in our working lives. Yeah.
Amazing. Thank you. Hortensia?
I guess for me, the thing that I learned, and this is when I started doing co-op in particular. So I worked with dystonia for a number of years before I started working more intensively with kids on the things that they really wanted to work on. And I always say when I train therapists on the approach, I learned to do co-op from them. There were, and I think there's always an underestimation, particularly in children's services, about how much they can problem solve. And I think the solution, it is the children and young people with dystonia that have the solution. I don't have dystonia. I don't know how to carry a cup of tea. I can't, it is them that can figure out the solutions. And I think that I always say, to me, the people that have the solutions to these kind of problems that OTs wants to solve is the children, young people with dystonia. That's what I learned from them.
Amazing. I mean, I'm sad that we have to wrap things up, but we do. Firstly, I hope you'll all join me in saying a huge thank you to both Anna and Hortensia. Amazing presentations, amazing answers to the questions. Thank you very, very, very much. Hortensia, Anna, thank you to my team, Bernie and Victoria. Thank you, guys. And for everybody that has stayed with us, listened, watched, and asked questions, thank you all very much, and good night.